Ibrahimi Warkani. He is from Iran, trained in different countries, Singapore, USA, now working in as a professor in Australia. And he is today willing to give a talk to the students in India as well as our scientist colleagues elsewhere. Dr. Majid E. Warkani is a professor and CINSW Fellow in School of Biomedical Engineering, University of Technology, Sydney in Australia. He received his PhD in Mechanical Engineering from Nanyang Technological University in Singapore. And he undertook postdoctoral training in Massachusetts Institute of Technology, MIT in USA. Dr. Warkiani is a co-director of the Australia-China Joint Research Center for Point-of-Care Testing and a core member of the Institute for Biomedical Materials and Devices and also Center for Health Technologies at the University of Technology, Sydney. Dr. Warkiani his current research activities focuses on three key areas: microfluidics, organ on chips, and 3D microprinting. Ladies and gentlemen, please welcome Dr. Majid Ibrahim Warkiani. Dr.
Thank you very much. Yeah, thank you very much for your kind introduction. So you all hear me well?
Yeah.
Okay, fantastic. So without further ado, I will start my presentation and to give an overview of what we are doing as an engineer in context of cell biology. And given the fact that most of the scope of the program is around stem cells, I tried to put some slides that relate to stem cells. But the tech that we developed are technically good for a variety of researches. And I try to showcase some examples for you. Just as a disclaimer, I'm a stakeholder in these startups that we spin out with my students. Again, I believe there will be a lot of biologists in-house, but for those who are not familiar with the field of microfluidics or they started to learn and know more about it, that's an very, very exciting field of research, which has boomed over the past decade, and a lot has been happening across the globe in life sciences and biotics, where people try to harness the power of microfluidics, which is the science of manipulation of particles and cells at minute quantities inside the microchannels for a variety of applications. And by using microfluidics, you get a lots of benefits. Again, there is a lots of opportunity to miniaturize your system. It does do a lots of cost saving in terms of the reagents that you're using. Parallelization and throughput is something that you can get from the microfluidics, and of course, ease of automation and building GMP grade products that you could utilize in stem cell manufacturing. I tried to pick up 6 examples for you of commercially available product that they all have a core microfluidic component inside them. For example, you might hear of Prodigy machine, which is built by Miltini Biotech for closed-loop GMP-grade manufacturing of the CAR T cells for cell and gene therapy applications. And there are microfluidic components like device or organ on a chip system, which I will introduce you more, like the Emulate Bio One, is microfluidic cartridges that you're trying to mimic the physiology of the human lung tissues. And it had helped a lot during the pandemic for repurposing of certain drugs for the COVID-19. Or there are platforms like the Berkeley Light machines where you can use microfluidics to do the single cell analysis. And you can produce essentially a high throughput monoclonal antibody. I will do antibody discovery using these minute microchannels where you have opportunity to use lights to essentially manipulate cells inside the pockets and do a lot of cool things like T cell potency assays, looking at the metabolomics of the cells at the single cell levels, looking after different CHO cells or HEK cells to pick up the best clone for certain secretion profile, etc. Or a more recent one is these products came out of the Cambridge by 2 of my colleagues called Nuclera, where you have the opportunity to use digital microfluidics for cell-free protein synthesis on demand, or even next generation PCRs, which doesn't need crazy equipment. We call them home-based type PCRs, where you use isothermal amplification within microchannels to could do home-based testing. Again, post-pandemic, these things become so popular, and there are many successful companies are pushing boundaries using these platforms, or even platforms that you can utilize for rare cell sorting and cancer diagnostic, which I will show you more examples similar to the rare site machine. As you hear, my group is essentially focusing on three areas of microfluidics. We spend a lot of time to build microfluidics for particle and cell separation, ranging from cancer cells, stem cells, algae, yeast, and exosomes. We have been developing microengineered system using microprintings like micromixers and microfilters. For example, we are utilizing our micromixers to do DMSO mixing, very rapid mix and homogeneous mixing of the DMSO with the stem cells to enhance cryopreservation. Or we are building some functional tools like tumor-on-a-chip models or lung-on-a-chip models to look at different drugs, how they interact with the human tissues, and how we can essentially replace or complement existing animal models using these organ-on-a-chip systems. Again, you're all very familiar with these slides. We are all built of organs and cells. And when something go haywire, let's say your body develops a disease like cancers or autoimmune disease, a cell within an organ or a tissue or within our blood started to malfunction. And we often are in the need of isolating a subpopulation of the cells. And when you're working in a wet lab, you're always requiring to have some information about that particular subpopulation of the cells that you're interested, such as the size, density, behavior of the cell, surface charge, or antigen status. And giving the available information or the cell type of the interest, you either end up using flow cytometry approaches and FACS or magnetic approaches like MAX, or you might use microfiltration or density centrifugation as a gold standards. And again, all these systems in context of the stem cell therapy, which is the core theme of this conference, is specialized to build GMP grade products like these two, like one Sophia machines from Cytiva or Rotel from the Thermo Fishers, where you can do closed loop end-to-end enrichment of your cells for a variety of applications that you have in mind. In context of the microfluidics, we are essentially leveraging on same principles, but we try to shrink and downsize the way and the device that can be utilized for cell sorting. And again, over the past decade, lots of cool technologies using microfluidics has been developed. I just put an animation that shows six distinct subpopulation of the microfluidics approaches. We call them active and passive approaches that has been developed, and some of them are actually commercially available. So there are systems that use inertial microfluidics, like the machine that we built back in Singapore, and is commercialized by a company called Biolytics for cancer cell sorting, or platforms like the Curate Biosciences that they use DLD systems for T-cell purification, or Levitas Bio use the magnetic field essentially to leverage to do live and dead cell sorting or preparation of your tissue samples for the single cell analysis. Again, if you go to the literature and try to understand the principle and physics behind these platforms, you will find it very exciting, and you can, of course, think of developing new application using these scenarios. So my group, particularly since 2010, has been very interested on the concept of inertial microfluidics. Inertial microfluidics is the science of pushing and particles and cells within microchannels that they are in order of tens of micrometers to hundreds of micrometers in size and few centimeters in length. And we try to leverage on inertial forces, essentially. There are hydrodynamic forces that present in microchannels that we try to manipulate those forces in order to build a useful gadget for cell sorting. And again, if you are interested, you can read these review articles that we wrote. You can see people have come up with different type of geometries and shapes to utilize for cell sorting. And over the years, my group has built different versions of these devices using 3D printing, using injection molding, using the traditional soft lithography approaches for different purposes. And more recently, we are trying to utilize AI and machine learning also to enhance the productivity and performance of these machines, which are work in progress for new applications that we have in. So here, I'll give you a couple of examples of when we build these microfluidic devices, how we utilize them. What are the real world applications of them? The clear example is where we have been working for decades around cancer liquid biopsy and how we can utilize the power of microfluidics to go and find the residual cancer information in the format of either circulating tumor cells, clusters, exosomes, microRNAs, ctDNA using these technologies. And then we leverage on those informations to get some meaningful information about the cancers, both for the diagnosis and prognosis across different solid tumors. And over the years, a lots of format or different shape of this microfluidics has been introduced. And then again, in context of the liquid biopsy for CTCs, people for years have been taught that single CTCs are more dangerous, but they realize that single CTCs, they cannot be as tumorigenic as the CTC clusters, where they can carry more stemness characteristics with themselves. And nowadays, there is a lot of interest for community to isolate CTC clusters, where we believe that they are primary source of the metastasis. And even in a seminal study by Mehmet Toner group, they show that if you inject individual CTCs compared to the clusters to the body of the mice, the tumorigenicity of the clusters is 50 times higher than the single CTCs. And now we need a good microfluidics to could isolate them. And again, my group has come up with different variation and shape and format. These are 2 or 3 of these microfluidics that we developed over the years. The middle one shows a device with the spiral shapes where the blood cells from the cancer patients pushed down under the influence of hydrodynamic forces, while the cancer cells, which are bigger and less deformable, goes up, and in this fashion, we separate them. This is one geometry that developed 2014. This is the 2016 version. We had the 2020 version, and this is the most recent one that we developed in 2023, which is a hybrid version designed to isolate both individuals and cluster CTCs simultaneously. And again, to prove that this type of technology works, we have done a lots of clinical trials with different groups across the globe for different type of solid tumors, breast cancer, lung cancers, colorectal cancers, even glioblastoma. We have completed a recent study showcasing that this type of CTCs and clusters find the opportunity to escape the leaky vasculature in the brain and come into the circulation and cause the metastasis. Even for context of prostate cancers, we have showcased that you can also process urine samples. You don't need to often process the blood. Urine is a very good source of body fluids, but you often get a very large volume of the urines. And we showcase that you can leverage on the throughput of the inertial microfluidic systems and go and fish cancer cells from the urine of the patients with the bladder and the cancer cells. And more recently, since we started to do some andrology works, we realized that we can leverage on the power of our device to look for cancer cells in the seminal fluids. And we recruited around 20, 30 prostate cancer patients, and we showcased that even cancer cells find their way into the seminal fluids. But again, seminal fluid is a very viscous fluid, and processing it using the lab setting is very difficult to fish for cancer cells. But we showcased that the microfluidics, especially inertial microfluidics, can be a suitable technology for this purpose. And more recently, our works got covered in Channel 7s around the idea of utilizing this for child cancer programs, where you could even process larger blood volume, as high as 100 to 200 mLs through the leukapheresis process that can be isolated. We can push through these devices and search for the cancer cells. And again, isolate enough tumor materials to could run multiple assays, single cell assays, drug screening assays, and culture assay, or even generate xenograft models using the CTC isolated with the leukapheresis process. Another works through collaboration with colleagues, we showcase the possibility to utilize the same text for NIPT, not just for the cancers. Again, in screening of the pregnancy and complications of the pregnancy, cfDNA is the gold standards, but we know that you cannot get all the necessary information out of the DNA. So it's better to get the actual cells out. And we know that the standard procedure, CVS, is very invasive. That's why we started to collaborate with people to look for these rare trophoblastic cells that they come from the fetus to the mom circulation during the week 8 to 12 of the pregnancy, and we try to isolate these trophoblastic cells as a new modality for non-invasive prenatal screening. When you are working on the field of the rare cell analysis, there is a lot of interest these days around how we can utilize new generation of technologies, single cell genomics technologies to do single cell analysis, because it's very evident now that all the cells are not behaving the same. And the context of cellular heterogeneity in biology is becoming more evident. And in the field of stem cells, as you do research, it's more challenging now. So essentially, when you're culturing a population of the cells, as in mesenchymal stem cells, iPSCs, or hematopoietic stem cells, you cannot expect that all the cells within that population are going to do the same job, right? So that's why a new generation of technologies for single cell analysis has been developed over the years. And these are three categories of these devices that we have it here at UTS. But when we started to use them for the field of rare cell analysis, we realized that they are not useful because we always have a minute quantities of CTCs, and this system cannot process them. That's why two brave PhD in my group a few years ago started to work on a new science called static droplet microfluidics, where we try to leverage on a science of liquid segmentation in the hydrophobic surfaces to build a useful low-cost gadget for single-cell analysis. Essentially, the way that this microfluidic device works is that you are putting a PDMS slab that has microstructures on it on the surface, and you inject your enriched sample products. Then you can do a simple air sheathing to produce individual pockets and droplets of nanoliter size. And in doing so, you're essentially trapping individual cells or droplets inside these devices. And then the good things about this platform is that you can simply put it on a dry ice and generate frozen cubes, frozen picoliter cubes, where you have the opportunity to visualize under the microscopes and pick individual cubes and do the qPCRs or ddPCR on them. So we build a very simple workflow and low-cost approach using these microfluidics. And one of my PhD students is currently commercializing this new generation of static droplet microfluidics for single-cell analysis. But one of the cool things that we have done that you might find it interesting is you can run individual cells metabolomics screening. Essentially, the field of metabolomics in cancer in stem cell biology is pretty hot now. And one of the challenges in cancer biology that we have is that the expression of proteins like EpCAMs or cytokeratin can go up and down during the cascade of the EMT. And that's why we are looking for additional non-surface biomarkers to identify if a cell is a cancer cell or not. That's why we use the power of these devices. So essentially, you can imagine a tube that has few cancer cells and mixed with lots of white blood cells. You can simply inject those cells into these individual pockets. For this particular case, we fill up the channels with the glucose, and we know that the cancer cells consume more glucose compared to the normal blood cells. And we can look at the fluorescent intensity, like the pictures down here, and essentially, without doing any staining, we identify which pocket contain the cancer cells and which pocket doesn't contain the cancer cells. So again, you can think of doing this in a blind fashions and do the blood analysis and essentially quantify a cell that is a cancer cells and is metabolically active or not without even doing immunohistochemistry. They have been characterizing this device more recently, and we are running clinical trials. But again, the output of this system going to add additional layers to essentially do CTC scoring on top of the traditional immunohistochemistry or molecular analysis. And more recently, with the emergence of the technologies like digital spatial profilings, we are trying to build a version of this device where you can put a cocktail of antibodies and do hundreds of the proteins of microRNA staining on these individual cells using this DSP technology available by nanostrings. So as I said, these platforms is currently turning into a commercial products machine like this. And we're trying to essentially commercialize it in New York through a company called Decan Biosciences to could offer it to the clinicians and scientists down the road for both CTC and CTC clusters analysis for cancer diagnosis. On the same note of the cancer biology, they have been very, very fascinated and interested with the science of organ on a chips. Again, for those of you who are for stem cell biologists, you are very familiar with the organoid and the field of organoid research. Essentially, organoids can be derived either from the actual tissues or they can be derived from the iPSCs. And this type of cells can be mixed essentially with technologies like 3D bioprintings or microfluidics organ-on-a-chips to build more physiologically relevant platforms for drug screening. And to just tell you how fast and how big is this field become, this is the number of startups and companies that they are formed just over the past 6 to 7 years. All of them working of bringing one of these technologies to the market as an alternative or replacement to the animal models for a variety of disease modelings or drug screening application. So one of the good works that we have been doing is a works with a company in Singapore called AIM Biotechs, where we have been collaborating over the years to utilize the power of microfluidics to build tumor-on-a-chip models and essentially make it very high throughput so that you can do large-scale screens and assays. This is an example of early works developed by these technologies where scientists decided to co-culture the tumor spheroid at the middle of these microfluidic channels inside the hydrogels, and then again put on the one side of the channels HUVEC cells. This 3D confocal microscopy, the top one shows how influence of, for example, endothelial cells, the green cells, can cause phenotypic changes for tumor clusters like this one, and they are become invasive. Essentially, they're changing their phenotypes from epithelial to mesenchypals through the cross communication that is happening within this 3D microfluidic channel. This is pretty difficult to capture inside the 2D traditional Petri dishes, but it's very evidence that you can mimic this type of scenario of extravasation and intravasation using microfluidics. In doing so, I collaborated with some teams through the company called NovoCures to build tumor-on-a-chip models using these systems. And the way that this type of technology works is that this company is trying to harness the power of electric field to utilize it for stopping the cancer growth and mitosis. And they have a commercial product for the brain cancers, but they never tested the fundamental biology of that. And that's why we decided to build a mini brain on a chip for them. So essentially, it's a microfluidic device that mimicking part of the brain with embedded electrodes. So this is how they do inside the lab. Left side is traditional core lab biology, 2D cultures. They just grow the cells and apply the electric fields and show that it doesn't grow. On the right-hand side, we build more physiologically relevant models, 3D models where you have a gels and tissue, and you can co-cultures the healthy cells to look at it. If you're interested to read more about this technology, feel free to go to the NovoCure websites to understand what is the core value proposition. But again, the models that we built for them help them a lot to essentially bring these products into the markets as a new way of essentially treating the cancers in conjunction with the chemotherapy and immunotherapy. And if you go to their website, you can see now they have launched a lot of new clinical trials for a variety of solid tumors, harnessing the power of low-intensity electric field for the cancer management. In doing so, again, over the years, I've got lots of clever PhD students to come and work on different factors. One of other factors that is so hot these days in cancer biology is the role of tumor microenvironment and subpopulation of the tumor environment, including cancer-associated fibroblasts. So one of the works that we have been doing in collaboration with Broad is to see a drug called pirfenidone, which is designed for tissue fibrosis, and lung fibrosis studies can be utilized in management of the cancers. So that's why we have been trying to build lung-on-a-chip models or breast cancer models on a chips to build these 3D models and utilize the efficacy of these drugs in inhibition or reversing the effect of the cancer-associated fibroblasts in communication with the cancer cells. All these data are published and available. I just skipped through to just give you an idea how this type of devices help cancer biologists these days to better understand how drugs or combination drugs will interact with the cells and tissues. And again, our job as an engineer is to further develop and mature this type of technologies to could become routinely used across the globes in different labs and city, in different labs and countries, and more importantly, be a good replacement in usage of the traditional 2D models or traditional animal models. I skipped through this, but one of the cool things that we also recently completed a clinical trial showcasing the efficacy of the pirfenidone in conjunction with cisplatin for the breast cancer management. But I found that it is not just for the case of the cancers. I found one of my colleagues recently in Australia, Rebecca, utilize it to reverse infertility in women. So essentially, aging, which is the hallmark of infertility in the females, is a process that has component of tissue fibrosis. And they have shown that if you utilize pirfenidones, you can essentially reduce and make some women fertile again. And this is a fantastic study and endorsement toward the hypothesis that we have for the cancer biology. Again, we have utilized these models not for the cancers, we have utilized it for other malignancies or other application. In collaboration with Professor McClements, we have built preeclampsia models, a complication that is happening during the pregnancy, and we wanted to looking at the role of FGAB-PPLs as a modulator of the preeclampsia and how we can utilize new therapeutics, including extracellular vesicles and exosomes, to enhance or essentially treat patients with these complications. And or more recently, we have built more complex version of these with vasculatures to models tumor inflammations and see how we can use immunotherapy essentially in conjunction with mild chemotherapy to reduce the source of inflammations inside the tumors, or to reverse a cold tumors to hot tumors to enhance and facilitate the transport of the immune cells inside these devices. And again, there's a lots of nice colleagues here across Australia who are helping us with the stem cell biology. These are some brain organoids derived from iPSC. I'm sure you're familiar with that. We're trying to co-culture this iPSC-derived brain organoid inside our devices to enhance their maturation, to enhance the vascularization inside these iPSCs to could do much more functional systematic assays over a longer period of time. And this is the Holy Grail. Technically, we are trying to see how we can use existing bioengineering approaches, production of the prevascularized bed with organoid systems to build such a complex models and do, of course, head-to-head comparison with the animal models. This is the works that, for example, we are doing with the Professor Hansborough, looking at the effect of particulates or bushfires, which is often happening in the hot season in Australia on the lung and lung performance of the lung tissue inside the human body. Okay, so I decided to put a couple of slides for you around stem cell production and how microfluidics help with that. Again, for this audience, I don't need to emphasize how important is the stem cell research, but why we are seeing little progress in terms of the translation. And where is the bottlenecks? Where is the glitch? One of the biggest challenges that we have is the cost and associated cost for large scale production of any therapeutics. This is an example of a publication that we put the data around MSCs. For example, if you want to produce large quantities of the MSCs to could treat thousands of the patients, how many billion or trillion cells you need? And in doing so, you cannot use the Petri dishes or the traditional cell culture lab scenarios to do that. You need to utilize advanced models, bioreactor technologies to do that. But again, these technologies are either complicated or the cost is still so high, or innovation is needed that could reduce those associated costs through the production. Again, we have identified multiple problems, but one of the problems that we realize is that the bioreactors are typically using a traditional approach called microcarriers. Microcarriers are tiny microparticles that they are in the size of 100, 150 micrometers, that people try to use it as a substrate for adherent cells like MSCs or iPSCs to attach and grow within the bioreactors. So essentially, the benefit of them is that a simple 5-liter bioreactors can do the job of hundreds of thousands of the traditional plastic dishes to produce enough quantities of the MSCs. But existing microcarriers in the market has multiple challenges and issues, such as particulate breakage, clumping of the cells, and so many other adverse effects. So that's why in one project, we try to work with a leading stem cell company in Australia, and we try to see how we can solve some challenges, post-production, particulate removals, cell watch, detachment of the mesenchymal stem cells from the macrocarriers, and how we can get the power of the microfluidics to do that. So this is an example of one of those works, so showing that how three different type of microfluidics, essentially one macromixers, one cell separators, and one cell wash, can be fully 3D printed to build an end-to-end device. We call it Lego-like microfluidics. And you can simply connect that to a bioreactors to could do three jobs for you. The first component of this microfluidics is a macromixers that rapidly mix the cells attached to the macrocarrier with trypsin to separate them. So essentially, instead of leaving your cells with the trypsin for 30 minutes to could get the detachment happen, we can facilitate this in a matter of few minutes. And then the solution that contains macrocarriers and cells will come to a microfluidic device that I showed you before. Based on the size, we can separate the stem cells from the macrocarriers using this device. And in the later stage, we can send the entire complex, the cells, that they are separated to another chambers to do the cell wash with a fresh media and get rid of any other residuals and particulate, making the cells ready for cryopreservations and filling into the essentially microvials. So if you could imagine that you want to do this in the lab setting with traditional centrifugation, filtration, and all those stuff, you could have see how many steps that it involves. But we technically shrink multiple centrifugation and filtration into a small cartridge like this that could be connected to bioreactors for downstream cell processing. I'm not sure if those of you who are working in stem cells, you are familiar with the field of lab-grown meat or synthetic meat. There is a lots of research nowadays is happening to prevent animal slaughtering. And essentially, simply by taking biopsy from animals, you could produce enough cells in the lab to could produce synthetic beef, synthetic meat, synthetic chicken, synthetic fish, etc. And this field has been booming. But the core science inside here is, again, stem cell research and how you can scale up the production, how you can reduce the cost. So what we have done recently is that we built a company called the Smart MCs that use the power of droplet microfluidics. So we realized that there is a big opportunity for these macrocarriers, next generation macrocarriers development. And in doing so, and of course, doing the literature, we realized that there is not much eatable version of these carriers available. So we come up with an idea to use food-grade materials and essentially build these hybrid macrocarriers that have nutrition factor at the centers. They have a nice stiff substrate for these type of cells to could attach and a nice coatings that can facilitate the attachment and growth of the cells. And then again, to produce this type of macrocarriers, we leverage on microfluidics, droplet microfluidics to produce. Now we build in a scale-up version of these machines that can produce billions of these macrocarriers in a matter of hours. And we are now commercializing these things as an eatable version, which are biocompatible, animal-free, suitable for the food market. And the same version of that we are recently trying to use for mesenchymal stem cells and hematopoietic stem cells too. This is some result. This shows that some of these carriers, as soon as you dump the cells on top of them, in a matter of 5 days, cells will love it, attach to the surface and grow. And because the materials that you are using is eatable and digestible, you can simply digest them. So essentially, you don't need to do filtration and separation at all. You're adding some enzyme, which is cell-friendly. You quickly get rid of the macrocarriers and you get a 99% live cells population out of that. So in this way, we are crazy and in a factor of probably 20x reducing the cost of the larger scale cell production, especially for the food processing using this novel generation of the macrocarriers. Okay, so am I out of time or not? Did you get bored? I'm not hearing you. Do you like I give you one more example or not?
Go ahead, go ahead. No issue.
Okay, fantastic. The last part of my presentation, which is just few slides before we open up for question, is again to show you microfluidics can be applied in different disciplines. And in this context is for the case of assisted reproductive technologies or ARTs. Again, I'm always fascinated by the process of cell processing and cell separation. And a few years ago, I was talking to my colleagues who are in the business of IVF around how do you do the sperm separation and preparation and wash prior to the IVF. And if you go and check with any clinics or college, you see that there's still the two gold standard are either already density gradient centrifugation or swim-up approach. And again, there is a lots of data showing that first of all, human body doesn't do any of these things. Mother of nature doesn't select the sperm, these two fashions that we do here. And there is a lot of data showcasing that this way of doing sperm preparation can cause DNA fragmentation and damage, which the outcome of that can be poor embryo quality, low implementation rates, and higher chance of the miscarriage. And that's why the success of IVF has been stagnated at 30-35% over the past decades, and we haven't seen much improvement. Again, microfluidics has been helping a lot. And one of the cool things that you can do is go mimic Mother of Nature, mimic what body is doing, and then you could replicate the scenario in macro channels. And we have looked at how the reproductive tract system of females essentially do that, how we are doing it, and is there any way to technically mimic that inside the microfluidics? We recently wrote a review for Nature summarizing this finding, and we showcased that how different type of the IVF, such as droplet IVF, ICSI, and IUI bypass one of these natural approaches, and why we need better ways to do it. So this is an example of a new works that we have done in collaboration with the colleagues at Monash. We essentially build a microfluidic cartridge that has multiple steps of the filtration. The steps of the filtration is exactly what is happening inside the human body. Essentially, we look at the microstructures and microgrooves inside the reproductive tract system of the females, and we try to replicate that with the 3D printing and SEM images inside these channels. So you can imagine that raw semen can enter these devices in the outer circles. On the pre-filtration stage, the dead cells, blood cells, or cells that are not motile will left behind, and those that they have higher motility will bypass these microchannels, come to the center. Then we coat it here with a pack of beads that they are coated with certain biomarkers. Here, for this case, we use apoptotic biomarkers to get rid of those cells that they have apoptosis or going under apoptosis, and essentially at the centers you get a subpopulation of highly motile sperms with high DNA integrity. And this movie at the top bottom shows how this process happening from the left to right, and those green dots that you see that they are moving, they are sperm cells that they are transversing these microchannels, and this is the actual picture of these microfluidic chips that is produced. We have tested these things in clinical trials over the past 3 years, and we have shown that we can drastically increase the quality of the isolated sperm, especially reducing the DFI, which is an important metrics in IVF clinics. And my team put a short movie to showcase how easy it is to operate. Essentially, in the lab, traditionally, when they're using centrifugation, they need to do multiple pipetting and steps to make the sperm ready. So we shrink 2 of the steps that is happening in an IVF clinics into the 3 steps, and we reduce 2 hours time of the sample preparation to just 20 minutes by utilizing this technology. Please watch this 1-minute movie. So you can see clearly quality of the sample, the raw semen and the samples that is purified using this microfluidic ready for the IVF process. And again, we are trying to build more sophisticated version of that for the ICSI processings, which has the AI component into it, post-isolation. We can utilize artificial intelligence to do a scoring of the samples. I had many more slides to give you, but I had just 30 minutes to show you. So I tried to put the gist of the information that is relevant to this conference and community. I hope at least by now you know what is microfluidics and what kind of cool stuff you can do, and you could utilize it in your future research. I'm happy to take any questions if you have now. Thank you very much.
Yeah, sure. Thank you, Dr. Warkiani. Very nice and very elaborative, and too many aspects you talked about. It's beyond the capacity of the biologist in the early in the morning, I guess. Okay, coming to the point, maybe I will open up some questions maybe for you, and then I will hand over to the other participants from here as well as from other parts of India. Okay, so I will come to the last point uh, of your video, in which you have shown that the viable spermatozoa can be concentrated in the using this microfluidic system. Are you also approaching for sex specific sorting of the spermatozoa, concentrating the sex specific? For example, for animal, maybe the female animals are more preferred than the male, or maybe for beef industry, maybe males are more preferred over
That's
the.
correct. That's correct. No, sperm sexing is it's a very, very hot topic in animal industry husbandry. And again, depending on the type of application. So here, the idea is that we should not have any bias. We don't apply any bias toward X or Y bearing sperm. We want it to be, we are utilizing this for human, and we don't see any type of bias that this device is applying with this mechanism. This mechanism is exactly what body is doing. There are technologies that you can utilize for sperm sexing, and we are implementing one of those inside this microfluidics for animal market. Yes, for the cattle and equine, those type of industry, we are trying to implement and enhance the way that this device works, that you could have potentially sex the sperm at the output of the device.
Okay. I see in this amongst the audience, I see some people from Australia also is there. Dr. Ghosh is there. Arnab is there. Arnab, do you want to comment anything?
That's a really nice talk, Majid. And yeah, it's so informative. Yeah, I think we are planning for collaborations in future to do some work together, and we are writing grants as well. So yeah, I think we'll do some cool work soon with Majid.
Wonderful. Is there anybody online who would like to comment or ask any questions? Dr. Ghosh, anybody? I cannot see the names from here. From Bangalore, Dr. Ghosh.
Yeah, I think it's a very wonderful talk. So, as you said, that we are lost with the informations. But I have a couple of queries related to, like, when you use the micro carrier, like, for a mesenchymal stem cell separation, it is easy because you are using a plastic matrix over there. But for other cells, do you use antibodies, or what do you use for immobilizing the cells in the microchip?
For the case of macrocarriers
Yes.
and attachment to macrocarriers?
Yes, yes.
So typically adherent cells such as MSCs or CHO cells or HEX, they are naturally, essentially when you dump these macrocarriers to cell culture media, the surface is positively charged, which attract lots of fibronectins and protein from the cell culture media, which in returns enhance the attachment of the cells. They will have the first recognition. And as soon as the cells recognize the macrocarriers, they started to spread and then proliferate. This is the signals that they get, right? It's in nature of adherent cells in doing so. The type of cells that has been cultured has been fibroblasts, myoblasts, fat cells from fish, MSCs, iPSCs. These are the various cell types that we haven't tried using microcarriers.
And for exosome analysis, you do only analysis or separation of exosomes also?
So for the case of mesenchymal stem cells, we have two aims. One is the production of the cells themselves, how efficient at low cost we can produce them. But yes, that's part of our study is the byproduct of the secretion of these cells, which is exosomes and a small extracellular vesicles. So we are collect just the media. We don't care about the cells and carriers. We just collect the consumed media and go and do exosome isolation.
So, and then for trophoblast cells, could you separate from the blood vessels of the pregnant women, or what?
For the trophoblastic cells, we did this for the pregnant woman. It's a very, very, very tedious, difficult task because these cells are very, very rare in circulations, but they are there. We have tried probably 50, 60 pregnant women. The success rate was 25 to 30%. We could not find these cells in all pregnant women. But the volume of the blood that we were running, the peripheral blood, was around 10 to 15 milliliter. We realized that some companies now are processing more blood, like 50, 60 ml. If you process larger volume of the blood, you may find more of these trophoblastic cells. Yes.
And then for semen separation, what is the volume we have used for the microscope?
So this device can process up to 2 ml of the semen volume, and the collection is around 100 to 100 microliter from the center of the device, which is more than enough for both ICSI and droplet IVF.
Thank you. That's all I have from my side.
Dr. Ghosh, is there anybody amongst us, in the participant who has got any questions?
Dr. Dara,
Who is that?
this is
Bhure,
Sanjeev
Bhure.
Bhure from Bangalore.
Dr. Bhure, go ahead, please.
Yeah, very nice, Dr. Majid. Very nice talk. So I have one query. What would be the size in terms of nanometer or micrometer of your microfluidic system? My purpose of asking this question is, can we apply this for quantification of the virus particles?
Inertial microfluidics is suitable for micron-size particle, not nano-size stuff.
Okay.
But there are other technologies that they are available, microfluidics for nanoparticle modification and those type of works. And this particular tech that we are using, no, is not good for the virus application. It's just good for cell application.
Right. Yeah, thank you.
And last question I have is, what is the cost of those equipments and the chip?
Not so expensive. So we are typically produce this thing in our labs. The initial design and fabrication might be a bit expensive, but when you're producing them at a scales, just few dollars per device.
Some of our colleagues can collaborate with you to design that and help us build it up in India.
Yes, we are happy to collaborate. If you have any exciting application in mind or any particular questions that you think this type of text that I show you today is useful, they're all available in my website, so you can read those. All the publication is free in my website. You can check it there. If you find it useful, we can definitely sign NDAs, collaborate, and give some of these texts even to use in your lab.
Participants, you have any question? Who wanted to ask? Okay, if we have no questions, nobody, I just wanted to thank you very, very much, Dr. Warkiani. And this was really nice. When Arnav said to me that that you need to have him for your workshop, I did not know then, but I got to know you through him only. And afterwards, I looked at your websites and it was really amazing. And it was nice. Nice to have you on this workshop, and for the other 20 guys, 20 participants, those who are sitting over here. Maybe in their future persuasion of academia, they can contact you. Please accept from our sides. And thank you again for coming up, and have a lot of success. That's what we'd like to wish you. Okay. Thank you.
Thank you very much. Thanks for inviting me. Thanks for Arnab for recommending my name. I hope you like the stuff that we do, and this could be an opening for future conversation and collaboration. If you came to Australia for holidays, please visit us in Sydney. We will be happy